Hello everyone! Today I'm going to tell you about the role of cell interactions of PLG nanoparticles in the drug delivery life cycle. As you may know, the pharmacokinetics of nanoparticle-based drug delivery systems is generally evaluated by their plasma concentration time profiles. However, the blood partitioning behavior of polylactic co-glycolide nanoparticles, widely used in the formulation of doxorubicin and other anti-cancer agents, showed that a significant fraction of nanoparticles is bound to the blood cells. So they do not appear in plasma, resulting in misinterpretation of pharmacokinetic parameters. Therefore, the objective of the present study was to investigate the influence of cell interactions on the nanoparticle kinetics and tumor distribution. In particular, the nanoparticle interaction and uptake by circulating immune cells, such as monocytes and neutrophils, the so-called cellular hitchhiking, was suggested to play one of the key roles in nanoparticle deposition. For this purpose, intravitreal microscopy was used. For the visualization of both nanoparticles and released drug, doxorubicin, the nanoparticles were covalently labeled with Cy5 fluorescent dye, so that the fluorescence emission spectra of Cy5 could be well distinguished from that of the doxorubicin. Taking into account that drug release profile may significantly alter nanoparticle parameters as well as targeting capability, 3 different fluorescently labeled PLG nanoparticle prototypes were prepared. The so-called fast-releasing formulation and formulation with medium release, obtained by varying the pH of external aqueous media during nanoparticle preparation, and slow-releasing formulation obtained by the addition of counterion during nanoparticle synthesis that increases the retention of doxorubicin in polymer matrix. All nanoparticle prototypes exhibited an average size around 100 nanometer with slightly negative zeta potential. The drug release profile in vitro was investigated using 2 different techniques. The first one was performed using a simplified release media and a centrifugation method to separate nanoparticle-bound doxorubicin fraction, and the second one using dispersion releaser technology with a donor and acceptor compartments and medium replacement. The amount of the released doxorubicin was quantified using HPLC. Both techniques indicated a considerable burst release for fast-releasing formulation, the green line on the first and the second graph, while a prolonged release phase and lowered burst effect was observed for the medium release and slow releasing nanoparticle systems. In the current investigation, the doxorubicin and PLG fluorescence signal in the blood vessels and tumor region were monitored using intravitreal microscopy. Intravitreal microscopy is a powerful optical imaging technique for collecting dynamic data in vivo with high spatial resolution. In the present study, the 41 adenocarcinoma cells were subcutaneously implanted to biopsy mice, and real-time imaging was performed 10 days after tumor inoculation using inverted laser scanning microscopy and skin flap technique. Here you can see the nanoparticle distribution in the blood vessels after intravenous injection, and doxorubicin that is gradually released from the nanoparticles and accumulated into the cells of the peritumoral space. You can also observe it on the images below. The visualization of nanoparticle interaction with immune cells was performed after the injection of fluorescently labeled antibodies to stain immune cells such as monocytes and neutrophils. On the left 3D reconstruction, doxorubicin accumulation in the cell nuclei is well distinguished. You can see neutrophils on both images into the blood vessels, and the areas of nanoparticle extravasation, the areas of macro and macro leakage formations for PLG nanoparticles. Approximately 15 minutes after the injection, The unspecific adsorption of the nanoparticles to the blood cells, red blood cells in particular, is replaced by their redistribution to the immune cells that are able to capture nanoparticles and mediate their trafficking across the vessel wall to the tumor site. The tracing of neutrophils colocalized with doxorubicin-loaded nanoparticles highlights that nanoparticles were effectively accumulated by the neutrophils, the outlined regions of interest, and transported across the vessel wall. You can see this tracing in time and the corresponding video. The quantitative analysis of nanoparticle uptake by immune cells was performed by flow cytometry. The association of nanoparticles with subpopulations of blood cells was determined for both drug-loaded and placebo nanoparticles. Indeed, nanoparticle redistribution to the monocytes and neutrophils was observed starting from 15 minutes after injection and was increasing with time. To confirm the localization of nanoparticles in neutrophils and monocytes, confocal microscopy was used. It confirms that 30 minutes post-injection, a significant amount of nanoparticles has entered the immune cells. You can see nanoparticles right inside neutrophils. The circulation of nanoparticle prototypes was monitored for the first 45 minutes after injection by intravital microscopy. The extravasation through the tumor vessel wall via micro and micro leakages were detected for all nanoparticle types, enabling doxorubicin delivery to the cell nucleide's target site. The corresponding video you can see on the right. The leakages were characterized by their area, number, and spatial distribution. The profile of doxorubicin accumulation in the tumor area was assessed for 3 nanoparticle types. The significant difference in nanoparticle half-life were detected. You can see that fast-releasing nanoparticles half-life was about 80 minutes. That is significantly lower than for medium and slow-releasing nanoparticles. Moreover, it was determined that fast-releasing nanoparticles subsequently undergo agglomeration once entering the blood circulation, followed by their fast elimination. At the same time, all 3 nanoparticle types exhibited a similar mechanism of extravasation and distribution across the vessel wall to the perivascular space. To sum up, the nanoparticle uptake by immune cells appears to be an additional and very important phase in the nanoparticle drug delivery life cycle. Moreover, the medium-releasing PLG nanoparticle formulation appeared to be more stable in the bloodstream and exhibited a higher initial drug loading. Therefore, this formulation can be considered as an optimal formulation for nanotherapy. With this, I'd like to thank you for your attention.